This video is going to attempt to summarize the videos I have in playlist for Anatomy and Physiology 2, beginning with the chapter on the blood. Obviously, this is an overview, and the individual videos in the playlist playing in the background can offer some more details if that is something that you need. So first of all, when we were embryos, there was a point where the brain, it didn't work, the digestive system didn't work, the respiratory system certainly didn't work, but nevertheless, the heart was beating and pumping blood. It was the very first system to actually function, which, it indicates how important this is. What does this cardiovascular system do? Well, a couple of big things. One, it certainly helps to transport stuff. So, for example, the muscles of the leg, they need oxygen. That's coming from here, the lungs. They need glucose, which is coming from here, the small intestine. They need to get rid of carbon dioxide to the lungs. And so obviously that, belies a lot of transport. Hormones are made here in the pituitary gland that need to reach the gonads or the adrenal glands and need to go throughout the body. Certainly urea, the nitrogenous waste need to be eliminated. So this must be transported to the kidney to be eliminated. So certainly transport is a major function. Some other functions include simply the fact that our specialized cells are rather delicate, unlike, say, the hardy little microorganisms that might live in a pond. So they need to be surrounded by a fluid that has the same pH, the same temperature, the same ion concentrations. Otherwise, their homeostasis would be disturbed. The blood helps to achieve that. And then finally, there are things in the blood which protect us. So, antibodies protect us from infection, clotting proteins protect us from fluid loss. And so these functions of the blood were critical. Now, there's very little that we say about blood. What we typically do is break it into parts first and then speak about the parts. So if you were to spin blood down, or even just let it set, even if blood was just sitting, for a while, the heavy elements would sink. But obviously, if you're in a hurry, you can have a centrifuge or a microfuge which spins blood. At which point you find that there are three distinct layers. There is plasma, about 55% of the blood, which is cell-free. You have a thin buffy coat, buffy, being a term you can use for off-white in color. And then the bottom layer, the hematocrit layer, that's where red blood cells are located. And so before going into the cellular layer, perhaps important to talk about the plasma. So plasma is mostly water, about 92% water, but then there are other things in it. Certainly 7% of it is protein, and there are some various kinds. There are albumins, there are globulins, there are fibrinogens, and then each of these has different functions. Some of them transport materials. So plasma being mostly water, it is polar. But obviously there are things that are non-polar that need to be transported through the blood. Steroid hormones, estrogen and testosterone, for example, are non-polar, so they need to be transported. The proteins in the blood, as we'll see later, are important for osmosis. Water goes to where the solutes are, and these solutes, these proteins, are important in helping water, go where the body needs it to go. Globulins also include antibodies, which are immunoglobulins, and fibrinogen is involved in clotting. And so these plasma proteins, they perform a number of functions. So when we spin blood down, there is plasma, and then everything else is what are called the formed elements of blood. Now, I'd like to say blood cells, but as we'll see, platelets technically aren't cells, they're fragments of cells. And so I really want to say, on the bottom part is blood cells, but let's just use the term formed elements, which is a bit more correct. So there are these, blood cells and fragments of cells. Now, we make them all the time in a second. In that second, you may have made just, maybe up to 3 million red blood cells, maybe 4 to 6 million white blood cells, especially if you're fighting an infection. That's a lot of new cells. Now, in our body, we once had stem cells as embryos, which were capable of generating, different types of cells and new cells, but our body tends not to have that. So, for example, one of the reasons I hope not to have a heart attack is cardiac muscle doesn't divide. So if I have a heart attack and I lose some cardiac muscle cells today, if they die, I will have to get through the rest of my life with less. I don't get any more. But our blood cells, every second, we are making millions and millions of them. How is this possible? Well, that's because we have stem cells, which perform hematopoiesis. That's the fancy word for bone making, having cells differentiate to become the different bone cells. And they are pluripotent because these stem cells can develop into lots of different cell types. When we were embryos, well, our first blood was actually made in the yolk sac, which technically wasn't even inside our bodies. Then we made blood in our liver, which, was weird, but then also the red blood cells actually had nuclei, unlike the modern red blood cells we have as adults. A few other places could also make blood, like the spleen or lymph nodes. As once we were born, the only hematopoiesis occurred in the skeleton, but the positions of the skeleton varied. So as infants, pretty much every bone can have red bone marrow. But as adults, it's much more limited, as you can see here. There are irregular bones like hip and vertebrae bones, which have red bone marrow. There are flat bones, like the bones of the skull and the sternum. And in the limbs, it's really only the proximal end of the humerus and the femur that have the red bone marrow. Now, we also then have yellow bone marrow, which stores adipose. And if we were bleeding and had a period of blood loss, some of this yellow marrow might become red marrow in at least temporarily, to make up the difference. But after birth, we went from the point where every bone essentially has this red marrow, which can contribute to hematopoiesis. And marrow can exist in the spaces that spongy bone there. And so in addition to those cells here you see adipose cells in bone marrow. So this would be yellow marrow here. Now bone marrow is quite impressive. So it's just like a video where bone marrow goes and is moving across a microscope slide in the background. Because once again, as adults, we don't have that many stem cells. Most of our cells have already decided who they would like to be when they grow up. But when you look at the bone marrow, you see the opposite. You see stem cells, which are in the process of becoming. When given certain hormones, they will then go through a series of stages and become red blood cells. If exposed to different signals, they then produce white blood cells, or the cells which make platelets. So this video just goes back and forth. I'm sorry, it's not moving. And so the bone marrow is unique in that sense. There's a great deal of differentiation where cells are becoming different cell types in the bone marrow. So it's a special place of the body. In this animation, I kind of simulate some of that process. So each of these will start off with a stem cell, a pluripotent stem cell, which has the ability to differentiate into different types of cells. Now, if we give it one signal, so erythropoietin, for example, a hormone, that then tells this cell to now start turning on certain genes which will then change this cell into a red blood cell. Now, this will include a number of things, including ejecting the nucleus. And so it makes the protein hemoglobin and ejects the nucleus and the mitochondria, and then it becomes this red blood cell. So that bone marrow cell differentiated to become this red blood cell. But that was because we gave it the signal erythropoietin, which said we need more red blood cells. We could have given the same cell a different signal. There are a number of locally acting hormones called cytokines, and certain combinations of cytokines would then tell this cell that instead of making a red blood cell, we need not only to make a white blood cell, but a specific kind of white blood cell called a neutrophil. A different series of cytokines would have sent the command, we need a white blood cell, but a specific type of white blood cell known as an eosinophil. And the hormone thrombopoietin would have told this stem cell, we need you to become really big. A bone marrow cell called a METAMEGAKARYOCYTE would then fragments into 1,000 pieces, each of these pieces being a platelet. And so we send different hormones to these stem cells, which then guide their differentiation, telling them what the body needs them to become. Red blood cells are the most common formed element in blood. So if you look at this smear, those are all red blood cells, red blood cells, red blood cells. Oh, look, there's a white blood cell. Red blood cells, red blood cells. Oh, look, there's another white blood cell. Oh, look, here's a couple smudges of platelets. But everything else then is a red blood cell. Red blood cells compose more than 99% of the formed elements of blood. Now, not only is that a lot, that's a lot for blood, but nevertheless, but also because they're small, we could argue, I guess, a little about how many cells there are in the body. But we could estimate that maybe 1 in every 4 cells of your body is a red blood cell. That's an incredible number. So there are a lot of red blood cells in your blood. There are a lot of red blood cells, period, given the percentage of cells that they are in the body. And so every single second, we're making about 3 million of these cells, the most common formed element in blood. Now, as they form, they eject their nucleus. And as a result, then their mature state is what's called biconcave. All right, it's concave on both surfaces. It has ejected its nucleus, and that increases its surface area. So if you had something this shape and you were attempting to wrap it, say with wrapping paper, you would then have to push the wrapping paper into the depression. That would mean you would use a little more wrapping paper. So the surface area, the amount that you would have to cover with wrapping paper, is increased by having those depressions. You might say, big deal, it's a microscopic cell. Yes, but given the number of cells, which are red blood cells, if each one increases its surface area just a little bit, that is an incredible increase in the surface area that these cells have for gas exchange. So it is through the cell membrane that oxygen can go back and forth, carbon dioxide can go back and forth. And so this shape of being biconcave, certainly it is important there in gas exchange. The shape of red blood cells also then makes them more flexible, which is good because that means that they can bend through narrow capillaries, better than they otherwise would. When we discuss sickle cell anemia, one of the issues with that is that the cells are then less flexible. And so the red blood cells, they eject their nucleus as they are in the bone marrow. Rarely there are red blood cells which actually enter circulation, which still have their nucleus, which is about to be ejected. But in addition to ejecting the nucleus, then notice that the second largest organelle of the cell is mitochondria, and the red blood cells will eject mitochondria as well, which is important because mitochondria, this is the part of the cell where oxygen is used to break down glucose for energy. But the problem is that red blood cells are supposed to be transporting oxygen, carrying it from the lungs to, say, muscles. If they're using oxygen along the way, then they are being less, efficient as transporters of oxygen. And so therefore, by ejecting their mitochondria, they physically can't use the oxygen that they are carrying, which ensures then that they would be, good at transporting oxygen to the cells that need it. Red blood cells have a great deal of hemoglobin. This hemoglobin is being made in the bone marrow precursor cells before the nucleus is ejected. The nucleus would obviously have the DNA, with the genes for hemoglobin. And as a result, hemoglobin can make, say, a quarter of the weight of the cell. So maybe, 250 million molecules per per cell. This is actually rather impressive. So, just genetically speaking, a typical cell has a whole lot of proteins, but only a little of each one. Red blood cells are very atypical because there's one protein, hemoglobin, which makes up such an incredible amount of the cells. So that's really unique that such a large percentage of one cell is composed of one single protein. Now, by hemoglobin, this is specifically what I mean. We have genes for the globins, alpha globins, beta globins, and some others we use as fetuses. They attach what's called a heme group. The heme group is not protein, and it has a central iron. That's what this big dot in the middle represents. And so this is heme, and when we attach it to a protein, a globin, we then have hemoglobin. We have alpha globin genes, we have beta globin genes, and if you take two alpha hemoglobins and two beta hemoglobins and smush them together, you then get what we call hemoglobin. And once again, this is what we've been using since birth. As fetuses, then we have a few other things. So instead of beta globin, we can make, say, gamma globin. So hemoglobin binds oxygen. And you might not be impressed by that because don't a lot of things bind oxygen? Doesn't iron rust? Yes, it does. But hemoglobin reversibly binds oxygen. So in the lungs, hemoglobin will bind oxygen. But then when we send this red blood cell to a tissue, hemoglobin will let the oxygen go. It releases it. That's something that rust doesn't do. So it reversibly binds oxygen. So that's a neat trick. Now, it is the heme part of hemoglobin which binds oxygen. Some of the carbon dioxide, which is transported in the blood, is bound to hemoglobin, but it is the globin protein portion which can bind carbon dioxide. So hemoglobin actually helps in the transport of two gases. Oxygen binds to the heme, and carbon dioxide, a portion of it, can bind to the globin portion. Now, red blood cells have tough jobs. They are sent around the circulatory system maybe once a minute or so. They're banging around a lot. And if they are damaged, well, they no longer have a nucleus, which has DNA, which could then make new proteins, et cetera, to fix the cell. So as a result, red blood cells have a limited lifespan. An average red blood cell may live, say, 120 days. That's a problem, because that means that every single second, maybe 3 million red blood cells are dying in your body. That's a problem because this creates debris. So if in your circulatory system, every single second, there were 3 million more dead cells, eventually this would just clog your capillaries. It would block blood flow. There would be portions of your brain which would stop getting blood, portions of your heart, etc. That would be bad. So your body can't let red blood cells just die whenever. To the greatest degree possible, you have to control this death. And so what your body does is it tries to kill red blood cells which are nearing the end of their life so that they don't burst at random in circulation, which would create all of this debris. And so there are places of your body where white blood cells called macrophages then eat red blood cells. So most of your red blood cells will die because your cells kill them, perform phagocytosis of them. So particularly in the liver and spleen, we have areas where this happens. So for example, there may be narrow capillaries where older red blood cells, which are less flexible, then have trouble navigating. And these macrophages then pick off these cells which are less flexible as they have aged. If we so in the spleen, we split the spleen into two areas. There's the white pulp, which has white blood cells. Then there's also the red pulp, which has a great number of red blood cells, and where old red blood cells are being processed. In the liver, the macrophages can actually then have their own separate name and be called Kupffer cells. Here in this video, these Kupffer cells have been allowed to perform phagocytosis on a darker material. So you see them, as dark in color because they've undergone phagocytosis. So these are the cells of the liver, which can then perform the phagocytosis of old, worn out red blood cells. And so we want to destroy red blood cells before they have the chance to burst in circulation, and macrophages in some parts of our body manage most of this, like the liver and the spleen. Now, if we're going to be destroying that many cells and that much protein, obviously hemoglobin is a protein, we want to recycle it and be as efficient as we can. And so once we break down these red blood cells, the proteins are digested and the amino acids are recycled, just as you would do from the amino acids of your diet. But then the fate that follows the rest of it is kind of interesting. So we recycle the globin portion, and the video will catch up, I'm sure, in a second. And then the question then becomes, what do we do then with the remaining heme? Well, we certainly want to recycle the iron. Not only do we want to recycle it, we want to hide it so that a bacterium in the body, which is looking to start an infection, might be hard-pressed to do that if it can't find iron. And so the iron binds to a protein called transferrin. Not only can we now conserve the iron, but we can hide it from bacteria which would otherwise use it for infection. Now in the liver, this remaining portion can then be sent into bile ducts as bilirubin. Now, heme is pigmented. Obviously, blood has color because hemoglobin is a pigmented molecule. And as we break this down, it then is pigmented all throughout its processing. So bile is not transparent. Bile has a pigment in part because bilirubin is a pigmented molecule derived from heme, which was a pigmented molecule. As bilirubin from bile ducts then enters the small intestine, it then can be broken down there into two forms. If it's absorbed into the blood from the small intestine, it can then be excreted from the kidneys, where it passes as urobilin. This is what gives a yellow pigment to urine. Or if it remains in the intestines, it can then be processed by bacteria in the large intestine to produce stercobilin, which gives the browner pigment to feces. So as we break down all of these red blood cells, this pigmented heme then produces a number of pigmented byproducts like bilirubin, urobilin, and stercobilin. One important disorder, which is typically mentioned about here, is that the trait of red blood cells of being flexible and being able to pass through narrow capillaries is dependent on their shape. Now, because so much of a red blood cell is hemoglobin, genetic mutations which change the amino acid sequence of hemoglobin can then potentially change the shape of this protein. Because there's so much of this protein in cells, this can then potentially change the shape of the cell. And one common disorder is that where a glutamic acid, the normal amino acid, is changed to valine, which then changes the shape of red blood cells. And in their sickled, more rigid shape, they are a danger to clogging narrow blood vessels, where they could block blood flow in blood vessels in the brain and in the heart. Now, such a deleterious mutation, you'd think that this would be rather rare in the population. But because being a carrier for this trait does actually give one a bit of resistance to malaria, which is a a deadly disease which affects many warmer parts of the world, the frequency of the sickle allele and thus sickle cell anemia is more common than one would otherwise expect.